Hello and welcome to this biotechnology webinar brought to you by Nova Biomedical and Sartorius. Today we have a presentation from Kymab, a therapeutic antibody development company based in Cambridge, UK. The presentation entitled Data-Rich Cell Line Screening, Leveraging ambr Cell Culture and BioProfile FLEX2 Integration will be delivered by Rahul Pradhan, a senior scientist with over 10 years industrial experience in bioprocessing. Rahul currently leads upstream cell culture development activities at Kymab, which involves process development, optimization, and tech transfer to GMP. This presentation will provide a case study from Kymab, which shows how the integration of the ambr microbioreactor system and the FLEX2 cell culture analyzer can assist in improving antibody productivity, workflow efficiency, and reduce project timelines. My name is Royston Bowman, Applications Manager at Nova Biomedical, and I will be taking your written questions for the Q&A segment at the end of the webinar. If you have any questions for Sartorius, I'm also joined by Sunil Chatra, Product Champion for the ambr system. A recording of this webinar will be available on the Nova Biomedical website if you need to share it or listen to the presentation again. At this point, I'd like to hand over to Rahul.
Thanks very much for the introduction, Royston. So today I'll be talking about data-rich cell line screening by leveraging the integration between the ambr and BioProfile FLEX2. I'll start off by briefly talking about microscale systems in bioprocessing. Then I'll dive into the Kymab case study covering a manufacturing cell line selection campaign, and then conclude with a summary. Within the biopharmaceutical industry, various microscale systems are often used for screening and optimization studies, and these can include things like small shake flasks, pin tubes, plates, and scaled-down bioreactor systems. Now, limited sample volume is a key consideration for these systems. There has to be a balance between samples taken for in-run analysis versus preserving the vessel contents for product quality analysis. And there are significant challenges to obtain a full profile of offline data. Some clever sampling strategies are often put in place to get around these challenges, and things like sacrificial duplicate vessels are often used. Additionally, a combination of different analyzers with different sample volumes is often needed. For example, we might need a BGA for pH and gases, a nutrient analyzer for metabolites, and a separate cell counter for getting cell density and viability data. Also, further efforts are then needed to make decisions based on this offline data and then feed it back to the running experiment. All of this adds a lot of operational overhead in terms of the scientist's time, and also it has the higher probability of introducing errors. Now, the integration of the BioProfile FLEX2 with the ambr aims to resolve some of these issues.